Hi, and welcome to our lecture on the urinary system. We're going to be taking a look here at the gross anatomy and microscopic anatomy of the system that removes waste from our blood to produce urine. And we're going to begin here with this idea that there are only 4 gross anatomy components to the urinary system. As far as gross anatomy, at least the basics of it, it's really simple. We have the kidneys, which produce urine. We have ureters, which carry the urine down to the urinary bladder, which stores the urine, and then the urethra, which allows that urine to exit the body. So we're going to begin here with the kidney, and again, just recognizing that as an organ that filters blood and produces urine, and by filtering blood, we'll kind of talk about that a little bit later on as far as kidney functions. First, we'll address the position of the kidneys. So the kidneys are paired, so they are lateral to the vertebral column, roughly in the area from T12 to about L3. And we can see here that there's a little bit of overlap of the floating ribs protecting portions of the kidney, and there's a little area below that which is largely unprotected by bone, but protected by some muscles that go through that area, such as the quadratus lumborum. Then we have the idea that the kidneys are retroperitoneal. That is, that they are positioned posterior or deep to the peritoneal lining. That would be the parietal peritoneum, meaning that if you open up someone's abdomen and you move the intestines, you're not going to see the kidneys themselves. Instead, you're going to see lumps where the kidneys are behind some peritoneum, and actually the kidneys are also going to be surrounded by fat. Now we do want to recognize those other gross anatomy components. So the ureters or ureters are muscular tubes. They carry urine from the kidney to the bladder, so they are paired again, and they are going to begin at an area up here referred to as the renal pelvis, and we're going to exit the kidney at this concave side of it called the hilum, and then they will flow down here, crossing the iliacs just about at the point where the common iliac artery branches into the internal and external iliac arteries. Then that's going to enter the posterior side of the urinary bladder, this sac right here, which will store the urine. Now we want to recognize where that urinary bladder is located as well. So in both males and females, it is posterior to the pubic symphysis or symphysis pubis, and we can see that roughly here in the female. Here's where those pubic bones would meet in the middle, and that bladder is sitting immediately posterior to it. In men, the bladder is going to be superior to the prostate. And we see that here in a diagram with the penis down here. Here we have the muscular floor of the pelvis, and then the prostate is just up above that, and we have to go through the prostate to get to the bladder. Now this does have some ramifications because the urethra itself will then run through this prostate gland. You may be familiar with the idea that prostates sometimes enlarge, particularly in older men. This isn't always due to cancer. There's also something referred to as benign prostatic hyperplasia, which causes the growth of the prostate gland, and that may pinch off that urethra, causing some difficulties with urination. Of course, in men, this bladder is going to be anterior to the rectum, but if we take a look at a female here, immediately posterior to the bladder, we see the uterus. So the bladder is going to be anterior to the uterus and the vagina, depending on exactly which portion of the bladder you're talking about, the superior or inferior portion of it, in women, whereas obviously men don't have that uterus, so the only other structure sitting there within that true pelvis would be the rectum. So we can get a little bit of a view of this here if we want to kind of get an idea of positioning of things. Here's the bladder sitting down here, and we have the rectum sitting here posterior to that within this small pelvis of a male. If we take a look up here, we see the kidneys. The parietal peritoneum has been removed, as has the adipose capsule to make them easier to see, but we can see roughly where the vertebral column would be by taking a look here at the abdominal aorta. Of course, we remember the inferior vena cava is off to the right-hand side of those vertebrae. We're also going to address the lining of the bladder at this point. It's lined by transitional epithelium. So we can see where the lining would be inside of the bladder here. And here we see some of that transitional epithelium, reminding us that transitional epithelium is stratified and that it has these cells which have kind of this lack of shape. That is, that they can change their shape as they go through stretch. It does even mention here in this particular picture that this is a relaxed epithelium. It's not stretched because the bladder is not full at this point, but we are reminded that transitional epithelium allows that bladder to stretch while still maintaining this water, or in this case urine-proof barrier, to prevent any urine from leaking out from this area, largely because these cells not only are stratified, but they also have what we call tight junctions in between them. All right, and we have our fourth and final component of the gross anatomy here. That is the urethra. And the urethra is this muscular tube that carries urine from the bladder to the outside. And we can see some differences here between males and females. So here's the bladder in both cases. In the female, there's a relatively short urethra leading to the outside. We find this opening between the labia. You can see labia minus here is the singular form of labia minora. And so it's opening up into an area referred to as the vestibule. In the case of the male, we have to go through the prostate, through the portion of the penis that is in the body, and then through the portion of the penis which is out of the body. And this actually does have some ramifications. This does mean that females are more likely to have urinary tract infections that make it to their bladder, causing what we call cystitis or a bladder infection. In the case of males, that's less likely to occur because there is simply more urethra, and the opening of the urethra isn't really positioned next to anything else. Now there are some other ramifications. In this case, this is going to be maybe not quite as good for males, but we have some muscles that control the release of urine, and this is much like it is in the digestive system. We have internal and external urethral sphincters, and we can see those here. So we have the internal sphincter, which is involuntary, which is part of the bladder wall. And then we have the external urethral sphincter, which is going to be part of the urogenital diaphragm, that floor of the pelvis. And of course, in females, these things are relatively close together, and there's not much urethra that's going to be positioned past that external urethral sphincter. In males, we can kind of see something here where we have, of course, the prostate to go through, and then we get to the area with that external sphincter, and there's a lot more urethra here. And I'm just going to share a personal story from 9th grade because I had this fellow student who kind of put this predicament in what I think are really some incredibly eloquent words. So I was at a urinal, and my friend Nick Greer was next to me there. And as we were urinating or finishing urinating, he kind of looked over, which is always uncomfortable, of course, in a bank of urinals in a restroom, and said to me, "Don't you hate it when your dick leaks?" You know, and I looked at him and I thought, yeah, I do hate that. The thing about that is that it is insightful to our anatomy, even if maybe it's put in a little bit of a crude term. We have all this extra urethra here, which potentially has urine in it, and you can do your best to try to empty this out, but maybe it won't quite work completely because, of course, portions of this penis are actually in the body. Not all of this is outside of the body. Therefore, you have additional urethra that potentially has urine inside of it, and sometimes the sad thing is you may put yourself away and there may be urine left in there that might leak out even if you have potentially tried to drain that. All right, so that's going to take us to kidney functions. We want to think about, why are we making this urine in the first place? And the first two bullet points here are kind of getting to the main purpose here. We want to eliminate organic wastes. This would be things like urea, things like uric acid, things like ammonia. And we want to do this while conserving nutrients. We don't want to accidentally get rid of things like glucose or amino acids. So we need some kind of process that allows us to take the wastes out of our blood, but not to lose the nutrients. Now, while we're doing this, we actually are regulating other things that are associated with our blood. We're regulating blood volume or pressure. Now, of course, these two things are related, and we do have mechanisms to control how much fluid we have in our blood. But we also have some other mechanisms associated with the kidney that may more directly relate to immediate control of blood pressure through things like vasoconstriction. So we've got kind of more than one approach to regulating our blood pressure. Then we regulate our blood pH That is kind of making sure that our blood isn't too acidic or alkaline. Primarily, we're going to be getting rid of hydrogen ions so our blood doesn't get too acidic. And we regulate plasma ion concentrations other than simply hydrogen ions. So we can get rid of things like calcium. We can avoid getting rid of things like calcium. We can regulate sodium ion concentrations, which of course are probably going to have ramifications for our blood pressure. And then we have a couple of additional functions that aren't necessarily regulation like this. We synthesize calcitriol, which is going to be something that will help stimulate the absorption of calcium. And so this, of course, is kind of going to be important for us. We also can directly excrete certain kinds of toxins, much in the same way that we can excrete hydrogen ions. And so when we think about getting rid of organic waste, those are largely products of our metabolism, but we can actually get rid of other substances in our kidney as well. So let's go ahead and take a closer look at the kidney, beginning with the idea that the kidney is protected in some ways. And we can see that a little bit because we have the kidney appearing differently on the two sides. This one over here, which happens to be on the left side of our cadaver, is sitting within its adipose capsule. We're sometimes called the renal fat pad, and there are even some more technical names for a couple of layers of fat that surround the kidney. But you can see it looks like there's just a huge glob of fat over there, which would be deep to that parietal peritoneum. Once you pick that fat off, there's just going to be kind of a thin wrapper around the outside of the kidney. That would be the renal capsule. So the capsule is a somewhat translucent kind of grouping of collagen fibers that you can peel off of this. We might recognize this as being an adventitia, very much similar in some ways to what wraps around those blood vessels like this abdominal aorta, or around sections of our intestine like the duodenum, which are also retroperitoneal. Now, if we think about the kidney, there are a lot of different parts of the kidney which will help us explain its function. So we want to begin with the concave medial portion of this. This is the hilum. You can see how the kidney bends in here. Of course, we know this is the medial side of it because we have this entry of our renal artery and exit of our renal vein. And obviously, these are going to have to either come from the abdominal aorta or return to that inferior vena cava. So the hilum is just the concave section of this right across here. And then we open up into what we refer to as the renal sinus. That's this area that is highlighted in yellow inside this dashed line. And you can see inside that sinus, there isn't fleshy kidney parts like we have out here in the other couple of layers of it, but rather there are these tubes in here, and we'll learn the names of those in a little bit. There are gaps between the tubes which have blood vessels in them, and any open space tends to be filled with adipose tissue. So that sinus is kind of the non-fleshy portion of the kidney, which is again on that medial side at that hilum entry point, and is largely tubes and fat. The other layers are going to be a cortex and a medulla. We, of course, remember what those mean. Cortex is an outside layer. Medulla is a middle layer. So here is our renal cortex. And we can see that generally that is lightly colored compared to that more middle area that consists of a number of triangles. Now, there are extensions of this cortex right down here which go in between the triangles of the renal medulla. And so we refer to those as the renal columns. So renal columns are really within the renal medulla, the middle layer, but they are extensions of cortex-like tissue and largely are going to be pathways for blood vessels. So the renal medulla is this darker inner layer. It is the sum of all of the renal pyramids. Renal pyramids are the individual triangles here, and they are functionally important. They're going to appear striped because of the collecting ducts which run through them. And of course they are darker. This is where a lot of the urine drainage is going and where we might engage in a process we refer to sometimes as water conservation. And this does bring up kind of the actual functional unit. The actual functional unit of the kidney is a renal lobe. Now we'll get to the microscopic units within these that produce urine in just a moment. But what we've got here is an area of cortex, and we're going to have kind of the beginning phases of urine formation happening primarily in that cortex area, and then we're going to have some of that urine forming process take place in this associated renal pyramid. So we link the cortex and the pyramid that is associated with together as a renal lobe, and that renal lobe will produce urine and drip that into a tube here referred to as a minor calyx. Now let's take a look at a real kidney. You might be saying, that does not really look like what we were looking at in the diagram. And that's kind of true. We can clearly see the cortex on the outside. That's kind of clear enough. This one seems to have a lot of blood vessels in it that we can maybe see. We can see some pyramids. They're a little bit less distinct than they are suggested in the diagram and or seem to blend together in some cases. Now, the sinus maybe is obviously tubes, some fat, some blood vessels, so that maybe is a little bit more clear. What we want to realize is that kidneys, of course, are working your entire life. By the time you are a cadaver, your kidney maybe is, let's say, running short on time, and may have even been one of the reasons why you passed away. It's not uncommon to have kidney failure listed as one of the factors that was associated with the death of cadavers, sometimes because of other underlying conditions, and sometimes it's the kidneys themselves that are the problem. So let's take a look at these tubes that are going to drain the urine from this. Again, this is kind of like the gross anatomy part of urine formation and getting rid of it, taking a closer look just at kidney structure. So we've got our cortex, we've got our medulla. Together they are going to form a lobe if we have one pyramid and the overlying cortex. We drain into a minor calyx. You can see a minor calyx associated with each pyramid, and those join together to form a major calyx. We've got another major calyx over here on this side. When those drain together, we get this big funnel-shaped area here. You can see that it's wide at the top and starts to narrow down as it exits the hilum. That would be the renal pelvis. Once it narrows down and exits through that hilum, it becomes the ureter. So basically, we form urine in our microscopic structures within the renal lobe. We drain into the minor calyx, to the major calyx, to the renal pelvis, to the ureter, down to the bladder, and then when we urinate, we're going to have that go out through the urethra. So let's take a look at what this might look like. I think that this is a very cool way of taking a look at what's inside the kidney. We don't have any of the veins, but we do have preservation of the arteries with all of the small branches, which we see kind of branch out here into the cortex primarily. And then we have things like the renal pelvis. We have a major calyx, and we have multiple minor calyces up in here. So this particular cast of the kidney shows us nicely what this kind of urine drainage system is. Minor calyx to major calyx to renal pelvis to ureter. Now it's time for us to take a look at the nephron. So the nephron is the microscopic tubular structures in the kidney, things where we have millions of these that are producing urine, and they're going to include a couple basic parts, the renal corpuscle here, and then the renal tubules, and then they will drain into a collecting duct. So it's all of this stuff from here up to this little ball here. Urine formation begins in that corpuscle, and we modify the urine as we work our way through the tubule component of this. Just because we will ask many questions about different parts of this nephron, I might refer to it on exams as the microscopic urine maker. So just associate the word nephron with microscopic urine maker. Now we do want to recognize that there are three basic processes of urine formation that are going to occur within this nephron. We have a nice stretched out nephron here to help illustrate part of what's going on here. Filtration is the first part of this. That's going to happen here in an area referred to as the glomerulus and the surrounding glomerular capsule, which form the renal corpuscle. And we sometimes refer to that as glomerular filtration. We then move into our proximal convoluted tubule. Convoluted means that it is twisted. Proximal meaning it's close to the origin, which is the glomerulus. And we're going to get here primarily tubular reabsorption. So this is mostly in the proximal convoluted tubule. We reabsorb most of the water that we don't want to lose and basically all of the nutrients that we don't want to lose. We then move into the nephron loop. It's a site of salt and water conservation. We could think of this as part of the tubular reabsorption if we want, or focus on water conservation. This basically reclaims the difficult to reclaim water. And then we move into the distal convoluted tubule. This is an area that is primarily for active secretion, often referred to as tubular secretion, where we take substances that are in the urine. We actually call that from this point at filtration, as it passes through this tube, the filtrate, and we do that until basically it drips out of that renal lobe. But in this case, we're kind of taking things like maybe hydrogen ions if our blood is too acidic or whatever, and we are kind of trying to make sure that we can actually pump those into here and get rid of them. We can do some reabsorption, where in the case of reabsorption, we might actually try to get things out of our urine and push them back into the surrounding area where we can pick them up into the bloodstream. So there are a couple processes that can occur there. And then we move into that collecting duct, which is really mostly about urine drainage, but under the influence of antidiuretic hormone, we're going to change the function of that tube. And we can see that a little bit in here as we get an idea of what's happening in these processes. So filtration is going to take the fluid from our blood and leave the cells behind. That's what we mean by filtration. Large objects don't make it into the urine maker, and small objects do. So as we enter the proximal convoluted tubule, this percentage remaining in the filtrate we can just say is the future urine, is 100% for every object. In other words, the concentration of anything, such as sodium ions, is the same at this point as it is in our blood plasma. Now, we say, I want some of that stuff. So in this proximal convoluted tubule, we reabsorb things like glucose, amino acids, and any proteins that happen to move into the filtrate. Those are completely reabsorbed back into our bloodstream, so they are gone or down to 0% left inside the filtrate. In other words, we should not detect glucose in your urine. If we do, you probably have diabetes mellitus, and that's an indication that there was too much glucose in your urine for the proximal convoluted tubule to be able to reabsorb all of it. We can see here that sodium is largely reabsorbed as well. There are some changes here where we do some pumping of sodium to help us reclaim some water. And you can see that we actually reclaimed most of the water in the proximal convoluted tubule, but it gets really difficult to reclaim more, and that's what the Nephron Loop is for. It's not that it's reclaiming most of the water there, but that it is making a huge difference by reclaiming difficult to reclaim water. And without ADH, we kind of level off. Under the presence of ADH, we actually continue to reabsorb water back into our body, particularly in this collecting duct. So antidiuretic hormone, the hormone that makes us not pee, is going to allow water to move from the collecting duct back into our bloodstream. All right, so let's begin taking a look at this piece by piece. We're going to start with the renal corpuscle, that beginning section of the nephron. And we can think about this as being the filtering component. It has a capillary network inside of a cup-like surrounding portion. And we want to recognize what both of those parts are. We're going to begin here with the glomerular capsule. That is this area here. And you can see it leads to this proximal convoluted tubule, which has microvilli on it. That's going to help us recognize that section. So we're basically kind of a cup, but we're also this layer of cells which wrap around the capillaries. We actually recognize this glomerular capsule as having a visceral layer and a parietal layer. And you can imagine the capillaries underneath here being kind of equivalent to the heart. And of course, we had like our visceral pericardium and our parietal pericardium forming the sac. So it's the same basic setup here. Now this is more historically known as Bowman's capsule, but you might ask yourself, what is in Bowman's capsule? Is Bowman in there? And the answer is no. But if we ask ourselves what is in the glomerular capsule, the answer is the glomerulus. That's the name for the capillary network, which is underneath this visceral layer of the glomerular capsule. So we want to think about how urine is formed as we go through this, and this is going to involve two arterioles and the glomerulus in the middle of this. We bring blood in through the afferent arteriole. This one's a little bit larger in diameter than the efferent. We come into this capillary network. Again, the capillary network itself is the glomerulus, and then blood will exit through the efferent arteriole. Now, this is easy because we already know the names afferent and efferent because we learned those in the nervous system. Now, what matters here is that we do have a difference in the diameters of these vessels, meaning that we can bring more blood in to the glomerulus than we can take out of the glomerulus, meaning some of it's going to be forced through these little slits here in that visceral layer of the glomerular capsule into this space and then down into the proximal convoluted tubule. So we're essentially forcing things through the glomerulus under pressure. That's why we refer to the glomerulus as the capillary filter. And we want to take a little bit closer look at this because it's actually something that looks super cool under a scanning electron micrograph. This is a close-up of the glomerulus, and obviously this is a diagram of it down below here. We see a cell on top of here. This is the nucleus of the podocyte. Podocyte basically meaning this is a foot cell, has these little foot-like extensions. It's almost like it's a sea star or something like that. And they kind of mesh together. It's like if you were to take your two hands and spread your fingers out and move them closer together and allow your fingers to kind of mesh together, you can see that maybe there's little gaps in between your fingers, and that creates these things down here that we call filtration slits. So we want to think about how this works as a filter. Well, number one, the afferent arteriole is wider than the efferent. That's going to produce pressure, which can force fluid out of this glomerulus, assuming that we have some way for things to exit. Well, the capillary endothelium is fenestrated, so we have fenestrated capillaries in here, and as a reminder, that means they have little holes. So we have still a basal lamina wrapped around this thing, but we can kind of work our way through these little holes and then through that basal lamina so that small objects will be able to exit the bloodstream. Obviously, large objects like red blood cells and white blood cells and stuff like that, they can't fit through those little holes. So they're going to stay inside the glomerulus and stay inside our bloodstream. And then, of course, these podocytes create what we call filtration slits. If you can't fit through those filtration slits, those little gaps there, you stay inside the glomerulus. You don't get filtered out. You don't enter the urine. So large objects stay in the blood. Small objects are able to exit and enter the filtrate, and that's what the rest of this setup is going to work on and modify until we get the urine we want to get rid of. So we move from that corpuscle into what we call the renal tubule. Again, we can just remind ourselves the nephron is a combination of the renal corpuscle, which is the glomerulus and glomerular capsule, and then the renal tubule, which is the proximal convoluted tubule, the loop of the nephron, and the distal convoluted tubule. And of course, each section has its own job, and they are a little bit different. The proximal convoluted tubule is the one where most of the reabsorption takes place. It's this one right here that comes off of the glomerular capsule, and it has microvilli. So although it is a simple cuboidal epithelium, as most of this renal tubule is, it does have microvilli to facilitate that absorption. The second section is the Nephron Loop, or sometimes called the Loop of Henle. And this is going to be this part that goes down in here. This may dip down into the medulla. It actually kind of depends which kind of nephron it is on how far it goes down into the renal medulla. But you can see here that it has both thick and thin portions to this. So we kind of want to recognize the difference between those. The thick section is going to be another area, which we see down here, having the simple cuboidal epithelium, and it's going to transport ions. So it's going to transport sodium and chloride ions into the medulla. That's going to make that renal medulla be salty, or if we want a more technical term, hypertonic. And that's going to help to draw water out of the filtrate. And where is it going to pull the water through? Well, the easiest place to do that, this thin section of the nephron loop, which is composed of a layer of simple squamous epithelium. So you can see those squamous cells here. So basically, these thick areas are going to pump salt out here. Water follows salt. Once we have this hypertonic or salty area out here, water wants to exit this thin section of the nephron loop. We can then pick it up inside of our blood vessels. And then we have our final section, this distal convoluted tubule. Weird thing about this, it does come right back by our afferent and efferent arterioles, so it is close to the renal corpuscle, but not as far as the actual process of movement of fluid, because it's very far from the origin of that fluid here at that glomerulus. So the role of this, again, is to actively secrete ions and acids to get those out of our blood and into the filtrate. This allows us to actually concentrate certain substances in the filtrate, that future urine, and it can selectively reabsorb things like sodium or calcium or water depending on the situation. Like that collecting duct, this is going to be a little bit permeable when it has antidiuretic hormone to be able to get more water out of the urine and back into our blood supply. You can see it's just a simple cuboidal epithelium, but it's lacking in the microvilli that we had in the proximal convoluted tubule. So how do we get the urine out of here and into that minor calyx? We begin by having that distal convoluted tubule come up into a collecting duct. The collecting duct can then run down through the medulla. You can see there should be multiples of these, and they will even join together here to form a big papillary duct, which dumps out through that projection of the renal lobe that sits down in the minor calyx, which we refer to as the renal papilla. So basically this is going to work. We go through the nephron into the collecting duct. Collecting ducts join together as they head down towards the bottom of that pyramid, forming a papillary duct which drips urine out into the minor calyx through the renal papilla. Now it ends up that we have two different kinds of nephrons that are doing this. We have what we call cortical nephrons, and we have juxtamedullary nephrons. And these are actually really good names that help us. Cortical nephrons, like this one, are located almost entirely in the cortex. You can see that even most of this nephron loop is in the cortex. There's only a little bit of this down in the medulla. We need to kind of think about this medulla for a minute. It actually gets more hypertonic, more salty or filled with urea as we head deeper down into it, and that's going to make it easier for us to pull water out of these tubes and into an area where we can pick them up using some of our blood vessels. So that's where the juxtamedullary nephrons are so good. They're called juxtamedullary because this filtering component of it, the renal corpuscle, is next to the medulla. It's still in the cortex, but juxta just means next to. So this is next to the medulla, and it has a long loop of the nephron, which goes way down into the medulla. So these are nephrons that are specialized for water conservation. And indeed, we do look at that length of the nephron loop as the key way for discriminating between these two types of nephrons. And that brings us to our little friend here. Big difference between our little friend here, this one happens to be Merriam's kangaroo rat, is that kangaroo rats are desert adapted, and it ends up that humans are really not so much. We are adapted to a terrestrial environment, and we do tend to have more juxtamedullary nephrons with their long nephron loops than do mammals that live primarily in aquatic environments, like, say, maybe a muskrat or a beaver. But we're not truly desert adapted like camels, or even more so these kangaroo rats. These kangaroo rats have the highest level of juxtamedullary nephrons of any organisms that we know, and this allows them to do something rather special. A kangaroo rat does not need to drink water during its lifetime. Now this is really interesting because it has to get water from somewhere, right? And it's not really eating food that is always particularly high in water. They're often eating seeds. And the way they get their water is actually primarily through cellular respiration. You might be familiar with cellular respiration, how we take in sugars and oxygen and produce carbon dioxide and water. Well, they actually get to use some of this metabolic water because they have such efficient kidneys that they don't have to rely on liquid water, which is probably good because it's often not available in such environments. So how do we end up picking up this water that we're trying to pull out of the loop of the nephron, or these other substances like glucose and amino acids that we're trying to pull out of that proximal convoluted tubule? We pick them up and put them into capillaries which surround the nephrons. So these are nephrons shown. We have a cortical nephron on the left, a juxtamedullary nephron on the right, which are surrounded by capillary networks. So the peritubular capillaries are exactly what they sound like. They surround the tubules, and they surround the short nephron loops of these cortical nephrons. So these are cortical capillaries. They are paratubular because they surround the tubes. You can see paratubular capillaries surrounding the proximal and distal convoluted tubules of this juxtamedullary nephron here, but it has a different set of vessels surrounding its long nephron loop. We refer to these vessels as the vasa recta. Now we can think about these terms. Vasa obviously means vessel. Recta, like in our rectus femoris, rectus abdominis, this means straight. So these are the straight vessels, and you can see that these vessels go down the length of the nephron loop and back up to parallel the nephron loop. This is going to allow them to kind of work in a countercurrent exchange with this nephron loop to be able to pick up that water that we're trying to reclaim. Now this brings us to our final topic. This is the Juxtaglomerular Apparatus. Obviously, we already learned what juxta means. Juxta means next to. This is an apparatus next to the glomerulus, and we see the glomerulus over in here. This is a structure consisting of a couple of cell types, juxtaglomerular cells and macula densa cells. You'll probably use those particular terms and maybe these mesangial cells as well as you learn about this in your physiology class. But these cells are going to work together to monitor the filtrate and to regulate filtration rate. So that's the glomerular filtration rate, something they might actually record as a measurement if you're getting some blood tests done. And ultimately, this is going to help to control blood pressure. So again, the juxtaglomerular apparatus is modified cells of the distal convoluted tubule here, modified cells here of the afferent arteriole. It sits in this area and is bound together at that place where the afferent and efferent are entering and exiting from the glomerulus. And if we want to think a little bit about what this is going to do, we can take a look here at some of this mechanism. So we've got our macula densa here, and you can see it's sensing low fluid flow, low filtration rate, or low sodium concentration. This is going to cause those juxtaglomerular cells to secrete this thing called renin. Renin obviously coming from renal, of the kidney. So this is this hormone that it produces and introduces into the blood. Now the liver is constantly forming something called angiotensinogen. So this is floating around in our blood, but the renin is going to activate this. So there's an enzymatic reaction, and it converts this angiotensinogen to angiotensin 1. It moves to the lungs, where this is converted by angiotensin converting enzyme, or ACE, into angiotensin 2. Kind of a complicated process. You don't have to know this here, but you will definitely learn this in physiology. This angiotensin 2 is going to do what its name says. It's going to cause widespread vasoconstriction. Angio referring to these vessels, and tensin to tense. So it's vasoconstriction, which will immediately raise blood pressure. So this is a mechanism, because of the low glomerular filtration rate, to raise blood pressure so that we can go ahead and increase that glomerular filtration rate and go back to producing more urine. Now it ends up that this also is going to stimulate the adrenal cortex to produce aldosterone. And aldosterone stimulates sodium uptake. So this is going to cause us to reabsorb sodium, which will cause us to reabsorb water, which is going to cause us to raise our blood pressure. So it's a second mechanism for raising blood pressure. Angiotensin 2 also is one of the things that can cause ADH, or antidiuretic hormone production. And this causes something weird to happen. We didn't cover this in the endocrine system, but it causes aquaporins, channels in membranes that allow water to move through them, to move to the collecting duct, plasma membrane. This is going to allow water to exit the collecting duct and enter the renal medulla, where it can be picked up by blood vessels and returned to our blood supply. So as antidiuretic hormone suggests, it's going to make us not pee by allowing us to reabsorb more water. So the juxtaglomerular apparatus ultimately kind of is the trigger point for recognizing we have a problem and responding to it in multiple ways by raising blood pressure.